Still chapter 26, and this video is on acid-base balance. So pH affects all functional proteins and biochemical reactions. So it is closely regulated by the body. Normal pH of body fluids, so arterial blood is about 7.4, venous blood and interstitial fluid is about 7.35. Intracellular fluid has a pH of 7. And typically the blood pH is going to be somewhere between 3.75 and 3.45. I don't know what I just said. Sorry. It's going to be somewhere between 7.35 and 7.45. Acidosis is when arterial pH is below 7.35, and alkalosis is when arterial pH is above 7.45. Small amounts of acidic substances enter the body in food. However, most of the hydrogen ions are produced as a byproduct of metabolism. And this includes the following things. Phosphorus-containing protein breakdown releases phosphoric acid into the extracellular fluid. In the event an individual is undergoing anaerobic respiration of glucose, they're going to form lactic acid. Fatty acids and ketone bodies from fat metabolism, and hydrogen ions are liberated when carbon dioxide is converted to bicarbonate in the blood. The concentration of hydrogen ions is regulated sequentially by 3 mechanisms. We have the chemical buffer systems, rapid first line of defense, brainstem respiratory centers, this is going to act within 1 to 3 minutes, and then renal mechanisms, and these are going to be the most potent. However, they take a little bit longer, somewhere between hours to days to affect pH changes. So when we start talking about chemical buffer systems, we first need to talk about a little bit about acids and bases. So in the beginning, we talked about acids and bases are electrolytes, and by in the beginning, I meant like the first video, I believe, or maybe the second video. We talked about the fact that acids and bases are electrolytes, and electrolytes dissociate in water. Strong acids are going to dissociate completely in water, and they can dramatically affect pH. However, weak acids only partially dissociate in water and are effective or efficient at preventing pH changes. Weak bases dissociate easily in water and quickly tie up hydrogen ions. Weak bases accept hydrogen ions more slowly. And so that's what we're seeing here. Strong acid, hydrochloric acid. Put it in water, it's going to dissociate completely. A weak acid like carbonic acid, you put this into water and it dissociates only partially. Chemical buffer is a system of 2 or more compounds that act to resist pH changes when a strong acid or base is added. It will bind hydrogen ions if the pH drops, or release them if pH rises. And there are 3 major buffering systems: the bicarbonate buffer system, the phosphate buffer system, and the protein buffer system. Now, the bicarbonate and the phosphate buffer system act very similarly. And we're going to talk about the bicarbonate buffer system first. This is a mixture of, and I'm going to say some of these names, but I don't want you guys to get too hung up on names. So some of them I'll mention, and some of them, like the ones we've come across in the past, like this one, H2CO3, is carbonic acid. We've come across this in the past. So a mixture of carbonic acids and salts of bicarbonate. So HC3O with the minus subscript is bicarbonate. An example is sodium bicarbonate, which is a weak base. And this is going to buffer both intracellular fluid and extracellular fluid, but it is the only important extracellular fluid buffer. If a strong acid is added, bicarbonate is going to tie up the hydrogen ions and form carbonic acid. And that's what we see here. We have a strong acid with a weak base, right? So hydrochloric acid combining with sodium bicarbonate, and we end up with a weak acid, carbonic acid, and salt. And this is only going to cause a small change in pH. and particularly in particular in this example, a small decrease in pH, unless all available bicarbonate, which is known as the alkaline reserve, is used up. And bicarbonate is closely regulated by kidneys. If a strong base is added, it's going to cause carbonic acid to dissociate and donate hydrogen. So here we have sodium hydroxide, which is a strong base, added to a weak acid, carbonic acid, and we end up with a weak base and water. And again, in this particular example, the pH will only rise slightly. And hydrogen ties up the base. The example here is the hydroxide ion. And carbonic acid supply is almost limited. And this is because of the CO2 released by respiration, and it's subjected to respiratory controls. The phosphate buffer system is nearly identical to the bicarbonate buffer system. The components are sodium salts of dihydrogen phosphate, which is H2PO4 with the minus subscript, which is a weak acid, and monohydrogen phosphate, which is a weak base. The phosphate buffer system is unimportant in buffering plasma. It is, however, an effective buffer in urine and intracellular fluid where phosphate concentrations are high. So we have kind of the same thing. We have hydrogen released by strong acids is tied up in a weak acid. So we have the strong acid mixing with this weak base, which is going to give us a weak acid and a salt. And then strong bases are converted to weak bases. Same thing. We have a strong base mixing with a weak acid, and we end up with a weak base and water. We move on to talking about the protein buffer system. Intracellular proteins are most plentiful and powerful buffers. Plasma proteins are also important. Protein molecules are amphoteric, which means they can function both as a weak acid and as a weak base. When pH increases, organic acid or carboxyl groups can release hydrogen ions. When pH falls, NH2 groups can bind hydrogen ions. And hemoglobin functions as an intracellular buffer. This right here is an awesome summary of the chemical buffer systems. So we have the bicarbonate buffer system, which is a mixture of a weak acid and salts of bicarbonate. This is the main extracellular fluid buffer, also operates some in the intracellular fluid. We have the phosphate buffer system, salts of this weak acid and weak base. This is an important buffer in urine and intracellular fluid. And then we have the protein buffer system, and this is where some amino acid side chains can act as weak acids or weak bases, and this is going to be the most important buffer in intracellular fluid, also blood plasma. We move on to talking about the respiratory regulation of hydrogen ions. Respiratory and renal systems are physiological buffering systems. They act more slowly than chemical buffer systems, but have more buffering power. Respiratory system eliminates CO2, which is an acid, and there's the reversible equilibrium that exists in the blood that we talked about back in the blood chapter, which is this equation right here. CO2 and water, and then can get converted to carbonic acid, which then can get converted to hydrogen ions and bicarbonate. During CO2 unloading, the reaction shifts to the left, and hydrogen ions are incorporated into water. During CO2 loading, the reaction shifts to the right, and hydrogen ions are buffered by proteins. If PCO2 in the blood rises, hypercapnia, it's going to activate medullary chemoreceptors, which is going to cause an increased respiratory rate and depth. A rising plasma hydrogen ion concentration, acidosis, is going to activate peripheral chemoreceptors, This will also cause an increase in respiratory rate and depth. Both cause more CO2 to be removed from the blood, which is going to push the reaction to the left, which is going to reduce hydrogen ion concentration. Alkalosis, on the other hand, will depress the respiratory center. Respiratory rate and depth will decrease, and this is going to cause the hydrogen ion concentration to increase. The respiratory system impairment, or respiratory system impairment, causes acid-base imbalances. Hypoventilation, which means ventilating less than what we should be, will cause respiratory acidosis. Hyperventilation, which is like breathing rapidly, will cause respiratory alkalosis. Here are the questions for this video. And that's all I have. Have a good day.